Good evening, everyone. It gives me immense pleasure to welcome you all to today's webinar. On behalf of the Center for Scientific Research and Development, People's University, India. Welcome to yet another exciting edition of the BioConnect webinar series, a platform led for student by student, designed to foster meaningful dialogue and connect academic knowledge with real-world challenges. My name is Inzia Kadri, MC of this webinar, and I am proud to tell you that I am an BSc Biotechnology student at CSRD. Today's webinar focuses on the important theme of from venom to medicine, snake venom research and laboratory biosecurity. This theme is a highly advanced topic that showcases how one of nature's deadliest substances can contribute to life-saving medical advancement. Snake venom contains a variety of biologically active compounds that are being studied for their potential use in areas such as pain management, cardiovascular treatment, and drug development. At the same time, such research requires strict laboratory biosafety and biosecurity measures to ensure the safe handling of hazardous biological materials and to promote responsible scientific practices. This theme beautifully connects innovation, medicine, and laboratory safety in the field of modern biomedical research. Before we proceed further, I would like to extend my heartfelt appreciation to the leaderships of People's University, our respected faculty members, and the dedicated student team at CSRD, whose efforts have made this event possible. We are truly honored to have several distinguished personalities joining us today. I warmly welcome Honorable Professor Dr. Harish Rao, Vice Chancellor of People's University, Professor Dr. Subhash J. Bhore, Head of the Center for Scientific Research and Development and mentor for this initiative, Mr. Rishi Nirwan, Chairperson of the Organizing Committee, and most importantly, our guest speaker, Dr. Chu Hock Tan, Associate Professor of Pharmacology at National Tsinghua University, Taiwan. We sincerely thank all guests and participants for joining us today for this enriching webinar session. Now I invite the Chairperson of the Organizing Committee, Mr. Rishi Nirwan, to deliver the welcoming remarks. Mr. Rishi, over to you.
Thank you, Ms. Insia. Good afternoon, everyone. Our esteemed guest speaker, Dr. Chu Hock Tan, Honorable Vice Chancellor, People's University, Professor Dr. Harish Rao, our mentor, Dr. Subhash J. Bhore, sir, respected dignitaries, esteemed faculty members, and my dear fellow participants. Hello, my name is Rishi Nirwan, Chairperson of this organizing committee of this webinar and a proud student of MSc Biotechnology at Department of Biotechnology and Microbiology at Center for Scientific Research and Development, People's University, Bhopal, India. On behalf of the organizing committee, it is my great pleasure to warmly welcome you to the session 17 of BioConnect webinar series, a special student-led initiative organized by the students of Center for Scientific Research and Development, Department of Biotechnology and Microbiology, People's University, Bhopal, India. Today's webinar titled From Venom to Medicine, Snake Venom Research and Laboratory Biosecurity explores a fascinating and highly significant area of biomedical science that bridges toxinology and laboratory biosafety. We are truly honored to have as our distinguished speaker, Dr. Tan, Associate Professor of Pharmacology, School of Medicine, College of Life Science and Medicine at National Tsinghua University, Taiwan. His remarkable contribution to snake venom research and pharmacological science continue to inspire researchers and students across the globe. I would also like to express our sincere gratitude to our Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, for his constant encouragement and his visionary leadership. A special note of thanks to our mentor, Dr. Subhash J. Bhore, Head of Institution, Center for Scientific Research and Development, People's University, for his invaluable guidance, continuous motivation, and unwavering support in making this event a great success. I would also like to acknowledge and appreciate the efforts of my fellow student organizers. Their teamwork, dedication, and enthusiasm truly reflects the spirit of academic excellence and collaboration at People's University. With this immense pleasure, I once again extend a very heartily welcome to all the participants and our expert speaker. I encourage everyone to actively engage throughout the session as we look forward to an enriching, insightful, and inspiring discussion. Now, I would like to hand over the program to our MC, Ms. Insia. Ms. Insia, over to you. Thank you.
To formally set the stage for today's webinar, it is my honor to invite Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, to deliver opening remarks.
Thank you, Insia Kadri. Good afternoon to all distinguished guests, respected faculty members, dear students, researchers, and participants joining us from different parts of India and abroad. It gives me immense pleasure to welcome you all to the 17th session of BioConnect webinar series organized by the Department of Biotechnology and Microbiology, Center for Scientific Research and Development, People's University, Bhopal. At the outset, I congratulate the CSRD team and our enthusiastic students organizing committee for successfully preparing for this unique student-led academic initiative under the inspiring motto, for students, by students. Such initiatives reflect a growing research culture, scientific curiosity, leadership, and collaborative spirit among our students. Today's webinar, From Venom to Medicine, Snake Venom Research and Laboratory Biosecurity, addresses a highly fascinating and globally relevant area of biomedical science. Snake venom research has evolved far beyond toxicology and now contributes significantly to drug discovery, translation medicine, diagnostics, and therapeutic innovation. At the same time, laboratories, biosecurity, and biosafety remain essential pillars of responsible scientific research, particularly in the handling of biological materials and toxins. We are indeed honored to host our distinguished expert speaker, Dr. Chu Hock Tan, Associate Professor of Pharmacology at National Tsinghua University, Taiwan. Dr. Tan is internationally recognized for his valuable contributions to toxicology, venom pharmacology, and translational biomedical research. On behalf of People's University, I extend the warmest welcome and sincere gratitude to Dr. Tan for accepting our invitation and sharing his experience with our academic community. I am confident that today's session will provide valuable scientific insight and inspire our students and young researchers to explore interdisciplinary research opportunities in life sciences, biotechnology, pharmacology, and public health. I also appreciate the efforts of Professor Subhash Janardhan Bhore, Head of the Center for Scientific Research and Development, People's University, Bhopal, for continuously organizing meaningful academic programs that connect students with globally recognized experts and contemporary scientific needs. So thank you, Dr. Bhore, for providing this platform and an opportunity for the students to organize this webinar and participate in it. Our participants learning beyond classroom is essential in today's rapidly evolving scientific world. Webinars like this serve as excellent platforms for knowledge exchange, networking, and intellectual growth. I encourage all participants to actively engage in the session, interact with the speakers, and make the most of this opportunity. I wish you all a great immersive learning experience. With these words, I once again welcome everyone to this important webinar and wish the program great success. Now I will hand it over to our MP, Insia Kadri. Over to you. Thank you.
Thank you very much, sir, for your inspiring words. Dear participants and fellow students, we are now ready to begin the core session of today's webinar. At this point, I am pleased to hand over the session to our moderator, Ms. Asta Singh Chauhan, who will introduce our guest speaker and moderate the session. Ms. Asta, over to you.
Thank you, Ms. Insia. Good afternoon, everyone. Welcome to this 17th session of BioConnect webinar series. I am Asta Singh Chauhan, and I am proud to tell you that I am a BSc Biotechnology student in the Department of Biotechnology and Microbiology at the Center for Scientific Research and Development, People's University, India. It is truly an honor to serve as the moderator for today's session. For today's session, I would like to introduce our distinguished speaker, Dr. Chu Hock Tan. Dr. Tan is an associate professor of pharmacology, National Tsinghua University, Taiwan. He also served as an adjunct professor at the Department of Pharmacology, Faculty of Medicine at University of Malaya, Malaysia. Trained as a medical doctor in Malaysia, Dr. Tan later obtained 2 research doctoral degrees, PhD and MD, in pharmacology and toxinology from University Malaya. He has authored more than 100 scientific publications in the field of venom toxinology with an H index of 38 and over 3,000 citations. Dr. Tan currently serves as the chair of the Asia Pacific section of the International Society of Toxinology and is also a life member of IST Honorary Secretary of the Malaysian Society on Toxinology and a member of WHO Snakebite Envenoming roster of experts. With this brief introduction, it is my great pleasure to invite Dr. Tan to deliver his talk on From Venom to Medicine, Snake Venom Research and Laboratory Biosecurity. Dr. Tan, over to you.
Thank you very much. Thank you, everyone. So without further delay, let me share my slide. Please let me know if you have any problem in seeing the slide or hearing me. Okay, is a slide visible?
Yes, we can see it.
Great. Okay, right. So first of all, I would like to, of course, say a quick thanks to everyone, to Prof. Subhas and also People's University for the invitation. So it's a great honor to be here, to know you guys, to meet you guys, though it's online, and to share a little bit of my research background with you. Hope this is inspiring, okay, for everyone. Okay. So my talk today is on this theme that is on From Venom to Medicines. I will try to cover a little bit about the snake venom research, an overview of it, and also a little bit of laboratory security, okay? So let's begin with Venom, okay? So I think we heard about this Venom works, quite often, but actually what is in the venom, that is what a scientist should ask. So we know that the snake venom, some venomous snakes, they produce venom, and in the venoms you will actually find the main components being proteins, okay? So proteins are one of these macromolecules usually, and you can use simple experimental or assays such as sodium dissolved SDS page, okay? So this is kind of gel electrophoresis to visible the distribution of the proteins in the venom sample, all right? Okay, so what motivates us to study these proteins, specifically proteins from snake venom? I briefly listed down some of the main motivations, at least for my group, is that we first of all want to understand the diversity of the venoms, okay? Because snakes are so diverse, there's so many types of snakes, so many species of them, so the venoms are also inherently very diverse. So there's a natural history behind it, evolution behind it, and it has ecological and also medical impact. And so from the medical or biomedical background, we want to understand as well how venoms work, because it will let us, it will give us insight into pathophysiological correlation. That means you know how venoms can cause damage or toxic effects, okay? That is in the case of snake bite. And also, if you know the pathophysiological correlation, then you also know how clinically, the patients or the victims of snake bite is going to progress. Then, more importantly, is to identify the key components in the venoms for treatment purposes, okay? So this will come to the neutralization topics later, especially when you talk about antivenoms. And of course, another aspect or motivation why we study snake venom research is that we still keep, a hope that although snake venom seems to be something, causing damage, something that is toxic, but they are all bioactive molecules. Okay, so they provide novel templates of biomolecules templates that is carry very high potential for drug discovery, okay, and applications as well. So we have some very good example that is already available in the markets. Some are drugs used for cardiovascular diseases like hypertension and heart failure, and some are actually medicines for blood coagulation problems. Okay, so these are some of the the application prospect, okay, that also come under the umbrella of snake venom research. So the scope of research, when you talk about snake venom research, most of the time it involves these three big scopes, okay, the three big categories. That is biochemical or biophysics methods. And of course, nowadays, is very much driven by the omics technology. Okay, people talk about proteomics, genomics, transcriptomics, these sort of things. And for venom researchers, we come up with a term that is called venomics, okay? That means omics technologies specially designed for venom study, okay? So on the functional aspect, you may want to do various in vitro or in vivo or even in silico methods now. Okay, so that encompass immunological assay, pharmacological or toxicological assays. Okay, to look into the functions and also the bioactivities of snake venom proteins. Then the applications part, this is perhaps what we can talk about then regarding the role or the impact of snake venoms on humans' health, okay, or our life. Are they friend or foe? Okay, so they can be friend because you can make them into new drugs, okay? New drug discovery, for example. But they can also be something considerably bad. Okay, so in snake bite, it causes damage, it causes harm, and there's also a possibility of misuse where it became, something that is with a bad intention. Okay, so this is about the applications that also come under the snake venom research. So I'm just showing you a picture. This is my favorite picture. This is a snake, obviously. Okay, can you name the snake? This is a pig viper, all right, and it is green in color, but in real life, it's actually very beautiful because it has some bluish hints, okay, under the scale, right? So this is a kind of a type of endemic highland pig vipers that is found in Malaysia. Okay, so this is one of the pig vipers. Morphologically is so beautiful, and so is the venom as well. For us, it's beautiful because it's so diverse. It has got so many bioactive components inside that waiting for us to study. Okay, so when you look at, let's say, across the globe, or perhaps you just look at Asia alone. Okay, so there are more than 100 species, venomous species. Sorry, if you consider the non-venomous, it will be even more. So if we talk about the venomous species, there could be easily more than 100, and all of them, are so different. They're so diverse in terms of the morphology, in terms of their habitat, and their, the ecological niche. But that also comes with variation in their venoms. Okay, so snake venoms, again, what are they? Remember, they are proteins, but they are not simply any proteins. They are proteins that has been honed by evolution to diverse into so many different types. Okay, so about the expression or also the translation of proteins, right, from genes to proteins, and because of such diversity, you have different types of proteins, and within the types you have subtypes of the proteins, of the snake venoms, and also each of these protein subtypes, they come in various expression level. Okay, it means the abundance in the venom is also different, and such variations occurs not just only within, not just between species, interspecies, right? Okay, it also can occur within a species. It means it's intra-species variation. Okay, and what is this protein or venom protein variation has got to do with health and medical practice? Well, it can cause a difference or variation in the biological activities. So the patients bitten by different species, of course, will come with different toxic effect, right? And also, therefore, the diagnosis has to be different because it's going to detect or affect the treatment and also the monitoring. And at the protein aspect, if you look at the antigenicity, okay, so because proteins come with antigenicity, which means how they can be recognized by the immune system or recognized by antibody, right? So it would affect or influence antivenom design. It's going to affect choice of antivenom. It's going to affect the dosing of the antivenom in treatment also, and that gives rise to different adverse effects because different antivenom, the doses, etc., will also come with different adverse effects. Okay, so just like for example in India or in South Asia, you have the very famous Big Four, right? Okay, so all these Big Four, they will require different antivenoms to tackle or to handle the different types of snake bite. Okay, and if you look at the geographical variation, from what I know, for example, let's say your Indian cobra, okay, Indian cobra from the south, from the east, from the west, or even from the north, they are different in terms of their venom potency and also the ability for antivenom to neutralize the different venoms. Okay, and lastly, the other concern or the other so-called is something that is promising, okay? Although these snake venoms are so varied, they are so variable, they have many variable proteins, but these proteins actually give us molecular novelty because it's going to guide us or help us for drug discovery. Right, so let me go through this some experiment aspect, okay? So this is some newest kind of at the moment, people talk about omics, okay, and also functional study to look into the protein variability. Because since I said, you know, proteins are so different from one snake to another snake. So what is the best way to look into these variations? Well, proteomics will give you the best answer, okay? So let's start from the scratch. Okay, let's say you have a snake here. This is a king cobra. Okay, a king cobra is you also have king cobra in India, of course. Right? So the king cobras, you produce a venom. So the venom you see is in liquid form, and you can lyophilize, means you freeze dry the venom. So this become a very stable and also relatively safe to handle materials. And these materials will be the substrate of the the sample, okay, the specimen for you to start your experiment. And snake, with of course with permix, etc., if they were euthanized, then you can actually get some tissues from them. So what's the difference of these two kind of specimens? If you've got the venom proteins inside, there are many, many proteins, okay? So this will be good for proteomics and all the other like peptidomics, toxinomics study after that, okay? Whereas if you look into the tissue, if you have the tissue, then what do you look for? You go into the DNA, right? You explore the DNA, you explore the gene expression, the RNA, etc. So this is where you have the venom gland, transcriptomics, and the genomics from the snakes. Okay. So together, you can call this venomics. Okay, so this is just a fancy word, okay, coined by our respected, one of the senior researcher in snake venom, Professor Guan. So he started this concept of venomics very beautifully. Okay, so back to our lab. Why do we study venom protein? Especially we look into the variation because we can, based on the, we look at the venom protein variation as a phenotype. You know what is phenotype, right? Okay, so as a phenotype of the species, and we can link it or relate it to the evolution or to the phylogenetics, okay? That is the basic science aspect. And beyond that, we look into the composition, so we can pin down what are the protein type, the protein different subtypes in the snake venoms. Then we can move on to study, for example, the reaction or the reactivity of the snake venom with antivenom. So we get things like antivenomics to look at specifically which proteins get recognized by antivenom. And we also can study the immunoreactivity, very easy assays such as Western blot, such as ELISA, to look at how good an antivenom binds to the venoms of interest, right? And of course, lastly is the neutralization aspect. For example, is the antivenom good enough to protect mice? Of course, yeah, that will be the easiest animal model from the envenomation of a snake. Great. So we cover the diversity, the structure and composition here, and then the medical or clinical aspect, which has the translational potential, that is about the indigeneity of the proteins, immunorecognition, and then treatment for snakebite toxicity. So this is why we want to study the venom protein variation. So I'll show you some series of a quick walkthrough for about the Cobra series that we have done in our lab previously. So cobras, to everyone, cobra is cobra, but for us, cobra is not just a cobra because there are so many species and subspecies, okay, within this huge complex. Okay, so some of the species that we have covered so far mostly are from Southeast Asia. And cobras, how do they kill? Well, neurotoxicity. Okay, yeah. So neurotoxicity, how do they work is because they block the postsynaptic nicotinic receptors. All right. So the transmission, particularly mediated by the neurotransmitter acetylcholine, gets disrupted, and you will have the victims will not be able to contract the skeletal muscle, and this is when then paralysis set in and patient die from respiratory failure. Okay, which can happen very quickly in within minutes to hours. Okay, so traditionally, conventional pharmacologists like to use this model where they take the nerve to get, this is a nerve and a bundle of nerve together with muscle, okay, from cheek, the biventral cervices around the neck, okay. So this is a human one, but we can't take the human sample, so we will use the chicken one, all right, the male chicken especially. So the example of how to study this neurotoxicity of the different cobras is that we set up the nerve muscle preparation. So when the skeletal muscle contracts in response to neurotransmitter acetylcholine, then the contraction will be detected, all right, by the transducer as a twitch, okay, as a twitch, which is like in a tension, right? A tension, okay. Then if you add in snake venom, like cobra venom or cobra toxins, then you can expect that these twitches will be abolished. Okay, these twitches will be flattened. Okay, and in vivo study, typically we use mice. So this is animal work where you inject the venom either intravenously or subcutaneous, okay, or intraperitoneal, and then you observe the quantum response, which means with increasing doses of the snake venoms, more and more animals will die, isn't it, right? Then you can calculate the LD50, okay, the median lethal dose from there. So this is some pharmacological kind of assay. If you're interested, I encourage you to study pharmacology, okay? Now, so an example that I'd like to share is one of our work previously done is that we compare this monocle cobra. This is Nayakautia, is a lot in Southeast Asia, and also in the northeastern part of India. Like I think Assam has it, right? Okay, so the northeastern part. Okay, but in this study, we concentrated on the sample, the geographical sample from Thailand, from Malaysia, and also from Vietnam. Okay, so the different geographical samples, you can see immediately from the chromatography. This is HPLC. Okay, you will see that this Thailand NKT, so Thailand sample, it has got this particular prominent peak, which we know and identify as the neurotoxin peak, okay, the neurotoxin fraction. So what is conclusion you can make from here, or the intelligent guess you can make from here? The Thai cobra definitely more neurotoxic, okay, more dangerous, even though they are of the same species. All right. So as I said, okay, the most stand up is this long neurotoxin, and it show, of course, you need evidence. So nowadays people talk about molecular evidence, proteomics relevant. You can use LCMS and things like that to help you to identify the protein and the protein subtypes. And then when you tested it on the chick by Venter, the chicken, the nerve muscle preparations, just now you can see that the venom of the Thai cobra depressed, okay, the muscle contraction very, very quickly compared to the other two. And in animals, of course, the mice die faster at a lower dose, okay. So this is what you can do from the nerve muscle preparation. Then, as I told you just now, we compare the time to 90% suppression of muscle contraction, okay? And since the Thai cobra has got more prominent neurotoxins, then the neurotoxicity potency in this case is stronger, much stronger than the Vietnam sample and also the Malaysia sample. Now, what about the neutralization aspect? So the assay or the experiment for snake venom research is that you can use the antivenom, try to reverse, okay, the paralysis, okay, in the muscle. So in this case, you see that for Thai cobra, because it is so neurotoxic, so the antivenom has got to increase. We need to increase the dose in order to overcome the neuromuscular blockade effect. Okay. Then another way of playing around this antivenom, rescue is that, to see how good it is able to rescue is that you put in the venom first and you observe for the muscle to, starting to draw or to starting to paralyze. Then only you add in the antivenom. Okay, the anti-MAV here refers to antivenom. Okay, so this antivenom is added in at different time points. This is exactly how you have in real life, clinically, right? So somebody got bitten at home or, in the village, and then they got transferred to the hospital. So there's a time lag in between, right? So there's a time duration or delay where the doctors in hospital is able to give the antivenom. So we observed that the longer it takes for antivenom to arrive, the worse outcome is going to be, isn't it? Right? So that means the early antivenom can be given, then you got better prognosis to restore the muscle function, all right? The muscle contraction function. Right, so another aspect of how to assess antivenom in neutralizing snake venom is to test it in animal model. So usually the animal model that we use is mice, okay? So the ICR white mice, the laboratory mice is the most commonly used. So first of all, we establish the LD50. So what is LD50? I mentioned just now briefly, median lethal dose. So it is the dose of venom required to kill half of the population. Okay, half of the population. In this case, is the mice. All right. So the lower the doses, the more potent, okay, or the more lethal the venom is. Remember that, right? Okay. So back to this graph, you see that Thai cobra has more long neurotoxin, and therefore, in the cheek by venter, the nerve muscle preparation, you see that the muscle contraction gets paralyzed quicker. And in mice, it's also correlate because the mice die at a lower dose of the venom, which means you need lethal dose of the venom, sufficient enough, is enough to kill the mice. All right. So the lower, the smaller the LD50 value, the more lethal, all right, the venom is. So why do we want to know the LD50? Because then we can standardize the LD50 for different venoms, and then we use antivenom to try to reverse or try to neutralize the venom. Okay, so in this case, we'll see that again because of the prominent neurotoxins in the cobra, in the Thai cobra, so the efficacy of the antivenom in protecting the mice from the challenge of the snake venom is much weaker, right, compared to the other two. That is because it has got more neurotoxin, the antivenom efficacy is low. Therefore, clinically, you can also see that the recommended dosage for for antivenom difference is different from country to country or from area to area. And the last one, okay, this is what we call most realistic, is mimicking the real snake bite. Okay, so this is called a challenge skill model, where you inject the venom into the mice and then observe for the animal to develop symptoms. Okay, so it's just like human. For humans, if someone is bitten by snake venom or something that is causing paralysis, usually the small muscle will get affected first. So for humans, typically we get ptosis, which means drooling of the eyelid. Okay, the eyelids will drop. Okay, so it's ascending, means the paralysis start from the top and then going down, right? But for animal like mice, we can't really see the eyelid to drop. Okay, but for animals like these rodents, they typically will start with a more general, weakness kind of symptoms. So that you can see is the posterior limb gets weaker, paralysis. They're dragging their feet, okay, in the cage. All right. So this is when then it indicates the neurotoxicity already starts, and this is also the time point where antivenom should be given. Yeah. Okay. So antivenom is given at this stage. So you will see that the antivenom, if it's working well, it will be able to reverse the neurotoxicity. So the mice will be able to recover with time. If the antivenom is not enough or the dose is too good, too low, then the mice will die definitely. Okay. So this is also another way how we can assess it. And then when it's back to the variation problem that we talk about that, then we also observe that the more neurotoxin there is in the cobra venom, you will need more antivenom, or even you have to give repeated doses of the antivenom. And the mice also take longer time to recover. Okay, so we expand the kind of study to different cobra, different species and so on. And we also got the honor to also study some of these Indian cobras from South Asia, including Sri Lanka as well. Then we sort of observe a pattern, all right, where you can see that the neurotoxins, remember just now we talked about neurotoxin is a type of the toxin, the proteins, but there are also subtypes. So there's a long chain neurotoxin, there's also a short chain neurotoxin. Okay, now you see that the long chain neurotoxin seems to be more prominent, okay, in the cobras from India, South Asia, okay, even to Thailand is part, all right. So Naya Naja Indian cobra, very famous. Thai cobra also very famous, and they're very, very neurotoxic, and they are mostly dominated by long neurotoxin. This is data from proteomics, okay. And as you move eastward, so the snake, of course, the cobra disperse more and more eastward towards east part of the world, including eastern part of the Vietnam, Taiwan, China, and then Philippines as well, and also the Javan Island. Okay, Javan Island is also considered like quite towards the east of the Southeast Asia. So these cobras, they begin to replace long neurotoxins with short neurotoxin. Okay, short neurotoxin. So in the Philippines, they even have like up to 40 to 60% of short neurotoxin. Only they don't need the long neurotoxin. And these two, both are neurotoxins, but their potency in causing neuromuscular blockade paralysis is different, and also their response to antivenom is also very different. Okay, so that poses a challenge to antivenom use later, especially when you're considering that you want to develop an antivenom that can, one antivenom for all countries, for all areas, for all species, then it becomes very, very challenging. Okay, so remember that, yeah? Okay, so this is just showing you that we have so many different types of antivenom. So even until today, we can't have one antivenom that can be used for all, right? Okay, it's because they are so different. Inherently, the proteins are different, and also the antigenicity are different. Then another group of snakes that I think could be very interesting and relevant to us, including friends in South Asia, is the kraits, right? So this is the Indian kraits, Bengallus carolus, which you have, very beautiful, the blue krait. And in the northern part of India and perhaps Pakistan, yeah, you have this Sind crates, right? The Sindh crates, okay, from the Sindh area of Pakistan, Bangladesh, Sindh. So we also studied that before, and we also compared it with the other crates from South Asia and also Southeast Asia and also East Asia. Okay, so the crates all, this is another crates. You see that they all have very special neurotoxin that is partly the basic phospholipase A2, PLA2, all right? So phospholipase A2 is actually an enzyme. We humans also have phospholipase A2, but the crates, they evolved this type of basic phospholipase A2, which we call beta neurotoxins, because it's going to target the presynaptic nerve fibers, okay, the nerve terminals of a presynaptic neuromuscular junction. If you remember just now for the cobra attacks the postsynaptic, whereas the snake venom phospholipase A2 here, it attacks the presynaptic nerve termini. Okay, so it causes respiratory paralysis also, and is usually resistant to anti-venom treatment. So it makes rescue and treatment even more difficult. But because the culprit toxins here mainly is a phospholipase A2. So phospholipase A2 is what is an enzyme. And we know that there are many inhibitors already available out there for phospholipase, for enzymes, isn't it, right? Okay, so beresparadib is one of the small molecule inhibitors that have been in the market many years ago. Supposedly, it's typically designed, developed initially to target systemic inflammation. Okay, inflammation including also like cardiovascular disease and so on. So it was initially designed to target our own human inflammatory phospholipase A2, but it has got discontinued clinically because of some side effects. Now, what people are trying to do is, what about, we try to bring back these sort of small molecule inhibitors and let's use it against the snake phospholipase A2, right? And it works as well, okay? So we are a lot of work currently going on using small molecule inhibitors to inhibit the enzymes or the proteins from snake venoms. So it's like, a kind of a new treatment, an emerging treatment that is independent from anti-venom because the mechanisms is different. Anti-venom works by having antibody binding to the antigen, right? The protein, all right? But if the antibody cannot recognize the antigen on the protein, then the antivenom will not work. But small molecule inhibitors, they have this beauty. They can, slot themselves, insert themselves in the cleavages and the crevices of the molecules, and then they inhibit the active site, for example, all right? Okay, so the active site usually are very conserved across different species, okay? That gives the small molecule inhibitors a higher chance to inhibit the toxicity of the snake venom. Okay, so this concept is called drug repurposing. Drug repurposing. So you repurpose an old drug or a drug that has been obsolete into a new drug now. Okay, it looks like a new drug for a different indications, such as in this case, people are trying to propose this beresparadib and the derivatives as inhibitors for adjunct and first aid use for pricks, so that you can save the patients even though he is far from hospital. He can receive this adjunct treatment and first aid, before he arrives at hospital. Okay, then quickly just, I still have time. Okay, just quickly talk about the applications now. Okay, so what are the, the good things that we can harness, from snake venoms? So I think you briefly have an idea now that snake venoms are so diverse, it has many proteins in it. And therefore, through this, the experiment biochemistry and so on, you will know where they are, all these different proteins. Then you can do the purification. You can even express them, okay, protein recombination. So you get the different key toxins. The different key toxin can be used to develop diagnostic, can be used to do drug discovery, can be used to develop next generation or new antivenom. Okay, so some of the toxin-inspired drug discovery, we have some successful example already out there. okay, like anti-cancer drug, analgesic, anticoagulant, okay, and experimentally, of course, we can always, depends on what species you're working on, you can always look back at your snake venoms and see what are the things that, potentially can be used to maybe to kill cancer cell. Then you have the anti-cancer pipeline coming in. And some of these proteins, they cause bleeding, right? So that means it disrupts the normal coagulation, the normal hemostasis. What can it become? It will become an anticoagulant, isn't it, right? And some of these, they actually block channels. So some of these channels involved in pain transmission. So they can actually become some sort of analgesic or painkiller. Yeah. Okay, and also because you see that the proteins are so different between certain species, then you may actually make use of these differences, and you develop venom detection tool, or in essence means diagnostics. Okay, so these are some of the examples that we have done before. We use one of the toxins that is very, you almost have it only in cobras. Then you can develop a cobra kind of detection tool. And also we use, we target the 4 main species in Southeast Asia. And then we develop the antibody, and also from there, then kind of rapid detection assay can be developed that is specific for the different species. So this will help clinicians to make decision later, when people are not sure what they're bitten, and then you don't have polyvalent antivenom, you need monovalent antivenom for better effectiveness. For example, this is when then the diagnostic kit can be useful. Okay. And for antivenom development, I think because there are so many different types of snakes, so if you can pull the important and critical and representative toxins from few species, then you may be able to raise an antivenom from horse, for example, that can work as a universal antivenom. Means it will target, you will also, you will neutralize, you'll be able to neutralize the toxins from the different species in that area. So in conclusion, for the first part is that I hope you are convinced that snake venom research is not scary, it's actually meant for good, okay? For improved healthcare. So we hope to have new treatment for snake envenoming, and we also hope that it will bring in new biomedical applications like drug discovery, for example. Okay, so the treatment approach forward, very well, neutralization. I mean, the treatment for snake bite always is antivenom, right? But also there's some other emerging therapeutics coming up now. Example, the small molecule inhibitions. Okay, so hopefully this will revolutionize the treatments for snake bite and venoming later on to save lives. Okay, just maybe a few more minutes. I just quickly go through this one is because we mentioned about biosecurity as well. So, the snake venoms, sometimes people so worried about that because they thought that is something that is harmful. So since I already mentioned that they are meant for good use, right? For scientists, it's always we want something good use, make good use of it. But you cannot be sure that there are some, people with bad intention, they might misuse. If they have access to the sample, they can misuse it as some sort of biological weapon and so on. But of course, I must admit that this is more relevant for infectious diseases usually, and for some sort of volatile toxins, nerve gas, that kind of thing. Snake venom is really unlikely. Okay, yeah, but I will still use this as an example. So I hope you today also learn about this concept about dual use potential. When we talk about dual use materials, it means these are stuff, things, goods, software, information, technology, whatever, that can be used for civil or military purposes. Okay, so some examples would be like nuclear, chemicals, microbe, toxins, navigation, like your drone and so on. Okay, these are supposed to be good, but it can be misused for bad intention. Right? Okay, so what should we do if you are dealing with materials, experiment materials that have potential, this dual use potential? Yeah. So first of all, you got to identify whether it is really, potentially harmful. Yeah, either to the environment or to human or to animals. Okay. So if you identify that it is really a materials with dual use potential, then the second things that you should do in our group is to do the risk assessment of the research, whether the benefit outweighs the risk to justify the research. Okay, and if the research is justified, the research must go on, then please mitigate the risk, minimize the risk as much as possible. So then you have a measures, and focusing on biosafety, biosecurity, and you got to make sure all the members are trained, they have good awareness, and you need to have periodic review of the research, communication plan, etc., etc. Okay, so this is risk mitigations, and it's not just about mitigation. You've got to make sure that the different levels, like institution, national, international, they have an oversight to monitor this sort of research, the material, and so on from time to time. Okay. So this will then justify and also make sure that the research is is meant for good, is for good intention, and then the security issue is minimized. So in summary, the first part we talk about the why we study snake venom, and then I also talk about the applications of why do we study, because we want to have better treatment for snake bite. We also hope that we can, make use of, or we can harness these snake venoms for diagnostic development, for drug discovery development. Okay, yeah. So this is the basics of what scientists do for snake venom research. But at the same time, we also share the concern for biosafety and biosecurity. So importantly is make sure to identify whether the materials is of dual use potential. If it is, then you have to follow the steps, okay, what to do to mitigate the risk and also the consequence from there. Okay, so with this, I thank you very much for your attention, and I welcome questions from the floor.
Thank you, Dr. Tan, for that highly insightful and thought-provoking talk on from venom to medicine, snake venom research, and laboratory biosecurity. Your presentation has beautifully highlighted the intersection of advanced venom research and cutting-edge biological models in addressing venom research. I am very sure our audience has gained valuable perspective on both the challenges and opportunity in this field. Now, we will move into the question and answer session. I encourage all participants to type their question into the chat box. We will do our best to address as many as possible within the time we have. To begin, let me ask you a few questions on the behalf of our participants. Could you elaborate on what was inspired you to enter the field of snake venom research and pharmacology?
Okay. My background is medicine, but then I sort of do my PhD study in pharmacology. And at that point of time, pharmacology is, full of cancer, diabetes kind of research. And I got this opportunity to do this very niche area of snake venom research, okay? That, kind of neglected, just like snake bite, it's neglected, but it's important. Okay, yeah, and I got fascinated by the diversity, and then I realized that, I never thought the same cobra can have so many different types of proteins. Okay, then I look into the functions and the structures, then I realized that it's really more and more and more, things for us to look into. All right, so that's why I got sort of fascinated by these snake venom proteins, and I think that we should continue doing research into it.
Well, that is indeed inspiring for many young researchers joining us today. Dr. Tan, during your talk, you highlighted the medicinal potential of snake venom components. Could you share which therapeutic applications currently appear most promising for future clinical use?
Okay, I think actually there are so many of them, and yeah, I just can't pick up one. But the most successful example, I think all students should know is ACE inhibitor. Have you heard about that? ACE inhibitor, okay? So ACE inhibitor, if you look at the history, it comes sort of, came from or inspired by the Botrops zaraka, which is South American pig vipers, okay? So these proteins serve as a template for the development of this successful group of anti-hypertensive, anti-heart failure, kidney protective drug until today. Okay, and I think that the cardiovascular disease, the drugs, there are still many more. Okay, to come into this. And other than that, also because of the metabolic, people are getting more and more. This is the more and more common disease globally, the metabolism problems. So how to reduce the stroke, cardiovascular thromboembolism kind of complication. That's also one thing, okay, that we should look into. So anticoagulants, antiplatelets, these are the drugs that should perhaps look into, Yeah. So there are many snake venoms that actually target this system, the coagulation system, the platelet system, okay? So actually, it's also a very good, example, okay? And I may just add another example if you want, is the GLP-1 agonist, right? The GLP-1 receptor agonist, okay? It's not from snake venom, by the way, but if you trace back the history, you can see that the very, very first template actually was inspired by a venom from Gila monster, which is a venomous lizard. It's a lizard, right? It's not a venom, okay? But you can see that, the drug is doing so well now, so for diabetes or whatever now, okay? So this is fascinating. So there are so many successful examples, and I think there's still a lot nature offers us, the snake offers us, waiting for us to dig into it, all right? Okay.
It is fascinating to see how substances once considered purely toxic can contribute to human health. We have question from our participant. Which enzyme is responsible for neurotoxin?
Okay. The most well-known enzyme responsible for neurotoxicity is the phospholipase A2, okay, the PLA2, right? So particularly in the Asian context, you have to think about crate, okay, the Bangala species, which you also have, we share, you also have in India, right? Okay, so crate, the presynaptic neurotoxin is phospholipase A2.
Thank you, Dr. Tan. We have another question that you also discussed the complexity of venom competition. How do modern technologies such as proteomics, genomics, or bioinformatics support advancement in venom research today?
Okay, so in the old days, people use traditional, more conventional methods such as chromatography, to slowly look into the diversity. But nowadays, everything, is high throughput. So proteomics helps very quickly resolve the complexity, the diversity down to the molecular level, the different types, okay? So that's a beauty of proteomics. And when you combine or integrate with genomics and transcriptomics, you get extra information about the genes, about the sequence, the nucleotide sequence, the protein sequence. And once you have all these, DNA things, you get the ticket to go into, the real differences from one species to another species, okay? So you get to do a lot more. So this is the beauty of the omics part. And bioinformatics, and of course, not to forget AI, okay? Artificial intelligence nowadays. So all these come in now, and they become like the in silico platform for us to help us, facilitate us to do sequence comparison, protein comparison, okay? And also help us to, within a short time, then we are able to see like where is the pocket, where is the active site, what molecule can dock, can bind to the pocket, and so on. So it actually greatly improves or facilitate our the efficiency, okay, in studying snake venoms. So I think the modern technologies really, really help a lot, advance a lot of this snake venom research.
Thank you for shedding light on the technological side of toxicology research. As laboratory biosecurity is an important aspect of this webinar, What are the most critical safety precautions researchers and institutions should follow while handling venom samples and related biological materials?
Okay. I think you have to treat the samples as something precious, of course, but also something that potentially hazardous. Okay, so if you think of it as a hazard things, you definitely will take good care of it. Okay, so the staff, I mean the students, whoever dealing with it, have to know how to, first of all, the PPE, for example, the personal protective equipment. Okay, so they must be trained on how to use it, especially for snake venom samples, and then in case of any spillage or whatever, what to do. Okay, although strictly speaking, it is less stringent as compared to virus, infectious agent, so on. Okay, less stringent, but we still have to take very good care of it. And then the log, like how you record the usage, all these have to be recorded, and then there must be periodic review by institution level, not just only the PI of the lab, but also some other institutional, council, whatever, they have to come in and, check or audit. Yeah. And then the samples usually, of course, the less people have access to it is better. Okay. So usually we will make sure that it's not like open to everyone. Any students can access. It's only in a few that has been trained. Then they will take good care of the samples. Okay, and other things like double lock and so on. Okay, the locking system and so on. So all these also have to be implemented. All right, in the lab.
Biosecurity awareness is especially important for students and early career scientists entering laboratory-based research. Dr. Tan, in your opinion, what are the major challenges researchers face in translating venom-based discoveries from laboratory research into approved therapeutic products?
The challenge, okay. So of course, as with any drug discovery or development applications from nature compounds, the challenge is always the I would say is that whether it can be used, in human. All right. So because snake venom, actually to start, yeah, it is something very, very useful, very powerful. Okay, very potentially, it can be used, but because it is inherently toxic, okay, so you will need to do a lot more to make sure that the benefit outweighs the risk. Okay, for example, like when you talk about like it kills cancer cells, yes, some toxins can kill cancer cells, but at the same time you must make sure that it doesn't kill the normal cell. Or yes, it kills the normal cell, but less, to the extent that it doesn't kill as much. And we must also establish that the efficacy or something, why is it like better than the existing drugs? There are so many anti-cancer, for example, out there, isn't it? So what is so special about these toxin derived anti-cancer lead that is possible, to develop into a new drug. Okay, so these are things that we will take times. It will take many, many years, and then it will need a lot of investment, a lot of money, to spend in typically more than 10 years or 20 years to make sure that it can be applied, it can be used in human. Okay, so this is one of the challenges that I think just as what we face with any other drug discovery situations, okay, that time is needed, investigation is needed, and also you need the resources, right? Especially investment to support the pipeline.
Well, that provides an excellent understanding of the gap between scientific discovery and clinical application. For students who wish to pursue career in pharmacology, toxinology, or biomedical research, what essential skill or areas of training would you recommend they focus on during their academic journey?
Okay, I think the basic must be good enough. I mean, I will encourage that you do well in basic sciences such as biochemistry, because it's proteins, genes, right? Okay, so biochemistry, and then pharmacology, if you can, all right? Okay, because it's going to talk about the interactions between the ligand and the receptor, whatever. So this is very relevant to snake venom work, okay? So pharmacology and also immunology as well, because some of these, like when you talk about anti-venom and so on, then you may also touch a little bit about immunology. So these are the basics one, but of course nowadays we have more advanced technologies. So the omics aspect, yes, you can also pick up, and also bioinformatics, because it will help you. It will facilitate your work, make you more efficient when it comes to like sequence comparison, handling large data, large sets of data, big data. So bioinformatics is also becoming very, very important. Okay. Yeah. I hope I answer your
Thank you
question.
for okay. Thank you for sharing such valuable career guidance to our participants. And we have another questions from our participants. As AI models can be used as predicting toxin structures, how can we balance open science sharing with the biosecurity risk of AI being used to design enhanced venom or anti-venom resistance toxins?
Okay, this is a tough question, okay, because yeah, I also don't know. Because it is, open science is such a things that people talk about now, and so you need to have a balance between like how much information you need to release into the system, okay? Because let's say if they want to design a new, enhanced venom toxin, anti-venom resistant toxin, this kind of things, they have to have access to the sequence, okay? The structure, the sequence, and then they modify from there. All right. Okay. So where will they get all these sequences and so on? It's all in the open database, right? Unless somebody has the sequence or has a structure, whatever, but they don't want to deposit it. They don't want to upload it into the open system. Okay. Unless somebody do that, right? But it's quite unlikely, because now everyone would want to talk about like, open science, and then you are supposed to share your findings, okay? So this is not very important. This is something very important, but it is not very easy to tackle at this moment, right? But if I can add on to it, I would just say that yeah, I think the balance is responsible, be responsible. So we have this concept about responsible open science as well. We should continue sharing the data, right? Okay, that with the good intention to help medicines, to help public health, like sharing, like let people know about the venom variation, so that the anti-venom producers, they know how to optimize the anti-venom productions, okay? But we should also be careful about information that gives direct instruction for engineering stronger or immune evading toxins. So perhaps the part about this is more about the technology should not be shared, right? So the whichever lab or companies, anti-venom producers, whatever, okay? So if they have the secrets, the technologies, like how to design or how to modify the structures, all these should be kept confidential if possible, all right, okay? So that although people can have the access to the sequence or the structures, but they may not have the very, very, the advanced technologies, as in how to engineering, the know-how, okay, the aspect that should be kept confidential. So maybe this sort of balancing will reduce the misuse of AI, okay?
Thank you for sharing such a great guidance to our participants. Finally, before we conclude this session, could you share your vision for future of snake venom research and its potential impact on medicine, biotechnology, and global health in the coming years?
Okay, big question. Okay, so I will answer a little. So so you're talking about the future of snake venom. Okay, so I think from what used to be very basic, now the trend of the research is moving towards more integrative, transdisciplinary, and hopefully it will become translational. So we will continue to discover venom diversity, the different venom structures, and then the different functions. And then we can also try to look into the how it can be harnessed for applications. Okay, so all these are still ongoing. So hopefully that with a more advanced technology, then like such as proteomics, omics study, structural biology, and even AI again, bioengineering, all these things, then hopefully we can design better diagnostics, better antivenom. If there's one antivenom that can be used for many, then that would be even better, right? Because antivenom supply is always a problem across the group. Okay, then the new emerging treatments such as inhibitors, that's also something that we should look into in the future. And then the new therapeutic discovery, okay, such as we talk about like new anti-cancer, new anticoagulant, all these things can also should also come into the future vision later. Okay, so hopefully these are something that we can make good use of in the future. And at the meantime, don't forget also, I think I haven't talked about this, but I also hope that, under the concept of so-called One Health, planetary health that people talk about now, actually snake venom research is also a very good, start point to remind us about biodiversity. Okay, yeah, and also I hope that it can let people know, awareness also that the snake venom, the diversity is not a source of danger or threat, yeah, but it's also a source of knowledge and for biomedical innovations to make our life better. Okay, I hope that is the concept that, everyone will see.
Thank you, Dr. Tan. Hello, participants. Do you have any questions? If yes, then please unmute yourself and ask questions. Alternatively, type your questions into the chat box.
Hello,
Esteemed
sir. I do have one
participants.
question.
Okay, sir.
Hello, sir.
Hi,
Myself, Rishi, chairman of this organizing committee, sir. I do have one question that this is very niche subject, and I do have multiple questions. But my main concern is that dealing with snake venom is a very legal and ethical bring challenges, and it is the most restrictive chemical in the whole world, and also the most expensive. So, in your research, how you overcome these ethical and legal challenges of every country? Obtaining the samples and then processing it.
Okay. I think there's some echo, right? Okay. So I think first of all, I'm not sure whether you say it's the most expensive. Yeah, maybe. Yeah. Okay. And then the most restrictive. Well, it may not as restrictive as some of the more dangerous, like some of the virus, and also infectious, and also some of these toxic materials, as in like bacterial toxins or some synthetic poison like gases and so on. Okay, it may not be as restrictive as this, but to overcome the whatever the the problem associated with the biosafety or biosecurity things is that you should trust the authority only. Okay, so when you come to like doing sample and so on, so team up with collaborators with the background. For example, like the universities researchers or their government researchers, okay, that work on perhaps snake venom, snake diversity, for example. So they have they know how to deal with these snakes, etc., and so on, okay. And from there, then they also know how to, like, for example, do the milking and so on. So you minimize the issue of about like wildlife, endangerment and things like that. Okay, so it's good, especially if you talk about like cross-country kind of things. It's always good to have collaboration. Okay, and then because the policy also regulation also vary from country to country. Okay, so it's good to first find out this from your collaborators from the certain different countries.
Thank you, sir.
Thank you.
Esteemed participants and fellow students, this has indeed been a remarkable session. I extend my heartfelt thanks to Dr. Tan for his valuable time and inspiring insights. I would also like to thank all participants for their active involvement during this session. Now, I hand it back to our MC, Ms. Cynthia. Over to you. Thank you.
Thank you, Ms. Asta, for completing the session effectively. To move forward, I now invite Mr. Hexuni to deliver the vote of praise on behalf of the entire organizing committee. Mr. Hex, over to you.
Thank you, Ms. Cynthia. Good evening, everyone. Respected dignitaries, faculty members, and dear fellow students. I am Chehak Soni, a proud BSc Biotechnology student from the Department of Biotechnology and Microbiology, Center for Scientific Research and Development, People's University, Bhopal, India. It is my honor to propose the vote of thanks on behalf of the organizing team and the Center for Scientific Research and Development as we conclude today's enriching session of the BioConnect webinar series. First and foremost, we extend our heartfelt gratitude to our distinguished guest speaker, Dr. Chu Hock Tan, for graciously taking time out of his busy schedule to be with us today. His insightful presentation on From Venom to Medicine, Snake Venom Research and Laboratory Biosecurity has been truly enlightening. We sincerely appreciate the valuable knowledge and perspective he has shared with us. We also express our sincere thanks to the Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, for his gracious presence and encouraging opening remarks. Sir, your continued support and guidance mean a great deal to us. Our heartfelt appreciation goes to all the respected dignitaries who join us today. Your presence has truly enriched this event. We extend our deepest gratitude to our mentor and guide, Professor Dr. Subhash Bhore. Your vision, guidance, and constant support have been the driving force behind this webinar series. Your mentorship continues to inspire and empower us. We will also like to thank our moderator, Ms. Asta Singh Chauhan, for conducting this session so effectively and ensuring its smooth flow. I would like to take a moment to highlight the essence of the BioConnect webinar series. For the students, by the students initiative, it stands as a testament to how passionate learner can achieve great heights when given the right platform to explore, question, and grow. This series not only connects us with global experts but also fosters a vibrant academic environment driven by curiosity and collaboration. To our enthusiastic audience, thank you for your active participation, engagement, and insightful questions. Your involvement truly added value and energy to today's session. Most importantly, we want to record a sincere appreciation to the management of People's University for their unwavering support and encouragement. Their belief in student-led initiatives like the BioConnect webinar series inspire us to dream bigger and achieve more. With these, I now hand over the proceedings back to our MC, Ms. Cynthia Kadri. Over to you. Thank you.
Thank you, Mr. Hex, for expressing our sincere gratitude to our respected guests, our expert speaker, and our esteemed participants. Dear participants, your feedback is extremely important to us. We kindly request everyone to take a moment to fill out the feedback form, the link to which has been shared in the chat box. Your valuable suggestion will help us to improve the quality and relevance of our future webinar. Now we will wait for 2 to 3 minutes to allow participants to complete and submit their feedback. We hope you have had a moment to share your feedback. Please note that your e-certificates will be sent to your registered email address shortly. With that, we came to the close of this insightful session of the BioConnect webinar series. We look forward to bringing you many more engaging sessions that connect science, society, and students. I'm Insia Kadi, signing off on behalf of the CSRD student team at People's University, Bhopal. Thank you all for joining us today. Wishing you a pleasant and inspiring evening.